right, gas transport. How do gases, okay, I'm sorry. We just talked about what affects gas exchange. And now I'm moving to the next question I have, which is about gas transport. How do the gases actually transport from the alveoli to red blood cells or from the red blood cells into the tissues? So, first of all, you pretty much know about oxygen already. You know that you don't know about this, but 98.5% of oxygen is bound to hemoglobin. So we know that the hemoglobin on the red blood cells carries the oxygen. 1.5% is dissolved in the plasma. Okay? And we know that, let me see if I can find my picture, that for one hemoglobin can carry up to 1, 2, 3, 4 oxygens. Okay? So, bound to hemoglobin, 1 to 4 oxygens for each hemoglobin. Okay, hope you can see that. What does oxyhemoglobin mean? It means that the hemoglobin has oxygen. Okay, that's pretty basic. The word itself says oxy. Where's my blue? So oxyhemoglobin is hemoglobin, which is signaled by HB, hemoglobin with oxygen. Now, it could be 1 to 4, what we just said. So right here, 100% saturation, what does this mean? That means that if it's 100%, every hemoglobin is carrying its maximum of 4 oxygen molecules. Okay? Deoxyhemoglobin, D, no O2. Now, when the first oxygen molecule comes along to bind into these spots, I can't remember why, but it's really hard for it to hook on. Golly, I should remember. But anyway, when it hooks on, that causes a slight change in the other 3 globular proteins, and so that the other 3 can go on. So basically, think about it. Here is a hemoglobin, all right? It has 4 spots for oxygen. The first one is sad because it has a hard time getting in and on. But once it hooks on, then the other 3 globular proteins, they're proteins, will change their shape and the other 3 come quickly, okay? And most of the time, if I were to check your saturation rate right now, it's probably around 99%, okay? So every one of your red blood cells will be carrying 4 oxygen molecules. So right here when I say, note the first oxygen to bind to heme is difficult. Once bound, the first one, the hemoglobin changes shapes, and it is easier for the remaining 3 oxygens to hook on. Okay, good. All right. I just want to tell you a side note. Right here, it's hard for the oxygen to hook on for the first one. but like exhaust from the car is called carbon monoxide. Carbon monoxide poisoning is easy because CO2, the red blood cell, has a 210 times greater affinity for CO than oxygen. So if you're breathing CO2, the red blood cells love it. 200 times. Their love is 200 times greater than that of oxygen. So therefore, when these guys start carrying CO to your brain, the brain hates it. So you're going to die. But anyway, I just find it fascinating that carbon monoxide, the affinity is 200 times greater than oxygen. Okay, moving on. All right, now, so this is kind of, for oxygen transport, is pretty straightforward. In carbon dioxide, it's a little different because it's transported in 3 forms. 3 ways we can get carbon dioxide into a red blood cell or carbon dioxide out of a tissue. So what are these 3 ways? Well, let me just look at what I've got here. Okay, so 70% of it, I like to use green for carbon dioxide. So 70% of CO2 is transported in the form of carbonic acid. But this carbonic acid dissociates quickly. So what I'm going to say. So this is a formula you're going to see quite often. You're actually going to see it again when we talk about digestion. But CO2 will readily combine with the water that's in a red blood cell. And as soon as it does, it'll form carbonic acid. And that right there, carbonic acid, that's the formula for carbonic acid. Okay. In order for carbon dioxide to hook on to the oxygen, we need a special enzyme. And that enzyme is called carbonic anhydrase, abbreviated CAH. Okay, so an enzyme, here you can write a side note somewhere. Enzyme equals carbonic anhydrase, ASE. And this is needed for CO2 and water to make carbonic acid. Oopsie. Okay, great. All right, as soon as the carbonic acid is made, though, it will dissociate into HCO3 negative, which this is a bicarbonate, plus hydrogen ions. Okay, this is very important. This reaction right here, very important. So that's 70%, 23% binds to plasma proteins in the plasma. And so CO2 plus HB is hemoglobin, will give us HbCO2. And these are called carb amino hemoglobin. Binds to, all right. So 23% binds to plasma proteins and to hemoglobin. Okay, so, and then if it binds to the hemoglobin, right there is what we end up. 7% carbon dioxide is carried in the plasma of the blood. So we can just say carried in blood. It's really the plasma as a dissolved gas. All right, so 70% that's transported from the alveoli to red blood cells is carried in this fashion. I'll explain this more in a minute. 23% is bound to plasma proteins or hemoglobin, and 7 is dissolved gases. Now, I don't want you to write right now. I just want to draw a quick picture and show you something, all right? Just watch what I mean. All right, so let's say that this is a tissue cell. I'll make it better in a minute, all right? So this tissue cell, we have carbon dioxide in here. And let's say that we have 70%. And here, this is a capillary. What's inside the capillary is a big red blood cell. Okay, I made it a little bigger, but I got a better drawing for you in a minute, all right? So what happens is the CO2 will come over. It'll hook up with water quickly, all right? Then it will dissociate and to make carbonic acid in the presence of CAH, the enzyme. This dissociates quickly into bicarbonate ion and hydrogen ions. Now watch this carefully. Now I'm going to redo this, so don't stress too much, okay? See, the problem is CO2 can't travel on the red blood cell exactly as CO2. It hooks on, but then it combines with water in the presence of carbonic anhydrase to create a carbonic acid. This carbonic acid dissociates into bicarbonate. This bicarbonate then gets into the plasma, and this bicarbonate wants to go to the lungs. It travels to the lungs, okay, to go to the lungs, okay? Because why? Because we want to get rid of CO2, okay? We just got it out of the tissues. We're traveling on the subway of the capillary to the lungs. And once it's in the lungs, we're going to get rid of the CO2, and that's what I'm trying to show you. So here at the tissue level, CO2 crosses, hooks up with the water in the presence of carbonic anhydrase, makes carbonic acid, which quickly dissociates into HCO3 negative. Now think about it. If all these negatives were to leave this red blood cell, you're going to create a polarity. It's going to be too positive in here compared to the outside, and that's not good. So how do we compensate for that? Well, in the plasma, we have a lot of chloride ions. These chloride ions, every time a bicarbonate comes across, hello, I'm coming in. Okay, so this negative that comes out, here comes a negative. And this is called chloride shift. Chloride shift. All right, and so that's good. Now, all this bicarbonate now, it has made it to the lungs. All right. So when it's in the lungs, the reverse happens. So what do I mean by that? So I'm just put this right here. All right. So here, alveoli. Remember, you're just watching. You're not worried about taking notes. You're just trying to learn, and I'm trying to show you here. So, and I'll draw this again. So, oopsie. So no worries. Shucks. All right. So, okay, here comes the blood. Yes, and here's a red blood cell. Yes. Now we're in the vicinity of lungs. We have a lot of this HCO3 negative coming in. All right. Once it's in the lungs, it wants to come back into the red blood cell. Okay. As soon as it's in the red blood cell, it wants to hook up with the hydrogen ion. When it does that, it makes carbonic acid, which dissociates into CO2 and water. Well, now we have a partial pressure really high of 46 millimeters of mercury of CO2 that wants to transfer across into the lungs. And then it goes up and out your trachea, out your mouth. All right. So basically, when you get to the lungs, you reverse the arrows and the reverse reaction happens. Keep in mind, if we bring a negative over here, Remember all these chloride ions in here? We're going to have chloride shift come out, okay? Because so chloride shift still takes place. So the bicarbonate, this is just the way the CO2 travels. This is the way the CO2 travels. So when it gets into the red blood cell, it hooks up with the hydrogen ions, carbonic acid, dissociates, gets rid of the CO2. Now let's look at this on your drawing. So you can focus a little bit on your drawing, and then I'll fill in the extra blanks. Look, you know what? See? 9, 10. I know you're dying, but you can take breaks. You can stop your video, walk around your house or whatever, and stretch your legs. All right, let's look at this. So here we go. What is this? You have this picture in front of you. Yes. Am I too high? There's my finger. So I think I'm still good. I have to make sure I keep on frame for you guys. Here, this is a legend to tell you what everything means, okay? So I'm only focusing on the 70% because it's the most difficult, and then we'll go on that. Okay, we're in the tissue cells. Carbon dioxide comes across in the presence of carbonic anhydrase. Right there, CAH, carbonic anhydrase, that's the enzyme. Here, H2CO2, H2CO is carbonic acid. We make the bicarbonate ion. It dissociates into bicarbonate and hydrogens, all right? Now, what does that say? I don't know what that is. Oh, Cl negative. It's just hard to make out. It's hard for you guys. So here's the chloride ion, okay? So the bicarbonate will come out of the red blood cell. This is how it's going to travel to the lungs, okay? Cl, chloride shift. You need to be able to understand the chloride shift. The chloride ion enters the red blood cell when a bicarbonate ion, HCO3 negative, leaves the red blood cell. All right. This is to keep the electrical gradient even, neutral. All right. All right. So great. So 70% goes that way, and that's how it gets out of the lungs. 23% hooks on to the hemoglobin. All right. So if you have carbon dioxide plus hemoglobin, you get, I think I've written it over here, carboaminohemoglobin. Okay. 7% will travel that way. 7% will just dissolve into the plasma, and it will, that's how it's carried to the lungs. The oxygen, we have 98.5% oxygen. What happens is, remember, the red blood cell has a lot of, let me pause for a minute. Okay. Start, play. All right, so here we have the red blood cell. This has a lot of oxygen. The hemoglobin has a lot of oxygen because it wants to give the oxygen to the tissues. So what happens is, this is a key thing. See these hydrogen ions, when this dissociated, these hydrogen ions will cause the hemoglobin to let go of oxygen, all right? So hydrogen ions cause the hemoglobin to let go of the oxygen. Now we just have O2, and it can float, 98.5% floats across. 1.5% is just dissolved. All right. This could be confusing. Make sure you just study. I'm trying to make it simple. I mean, not simple, but just try to explain it in a way that hopefully it makes sense. Last page. We can do this. All right. I don't want to get my pages messed up. All right. Okay, some notes you need to know. You were responsible for reading about oxygen imbalances such as COPD, smoking, lung cancer, emphysema. And you're also responsible, I think we left on slide 69 from our end of our lecture, second lecture, and you're responsible for the rest of those PowerPoints. I just wanted to say that before I forget. All right, adjustments. This is it. Once we finish this, I'm going to say bye-bye. Go eat some lunch. Adjustment to metabolic needs of individual tissues. Think about it. You're just sitting there. But if I ask you to get up and run, sprints for 20 minutes, then your muscles obviously are going to need a lot more oxygen. So exercising will cause the skeletal muscles to have a need for more oxygen. So what does this mean? So what are the 4 factors that adjust the rate of oxygen? So first of all, it's the pressure of oxygen, the ambient. So we know that active, maybe I'll do black. Active tissues consumes oxygen rapidly. Therefore, the partial pressure of oxygen in tissues decreases. Therefore, if the oxygen decreases, you're going to have oxyhemoglobin releases more oxygen. Okay, so let me refix it. So active tissues consumes a lot of oxygen. So now the oxygen levels in tissues is low, and this helps aid for the release of oxygen from the hemoglobin in the red blood cells that's in the capillaries. So therefore you can give up more oxygen to the tissues because there's a larger pressure gradient. Temperature, active, warm tissues, such as skeletal muscle cells in working, promotes oxygen unloading from the hemoglobin, unloading. Ambient pH, if you think about this, active tissues create extra waste, which is CO2. Therefore, remember, if you have a lot of CO2, then you're going to have a lot of hydrogen ions. So active tissues create extra CO2, therefore you have a lot of an increase in hydrogen ions being released. Because remember, CO2, because we have a lot of CO2, where did I put it? Active tissues creates a lot of CO2, so if you have a lot of CO2, it's going to hook up with the water, make carbonic acid, dissociate into bicarbonate to get to the lungs to get out, and then here you have a lot of these, all right? So if you have a lot of hydrogen ions, that will cause the blood to be more acidic. Okay, now remember, hydrogen ions are good, okay? Because what they cause, hydrogen ions, remember, hydrogen ions causes the release of oxygen from hemoglobin. And see, if you're working, don't you want to have a lot of oxygen available? Yes, all right? So hydrogen ions causes the release of oxygen from hemoglobin, and that makes your skeletal muscles happy. This is called the Bohr effect. Okay? So active tissues create extra carbon dioxide, got a lot of hydrogens, because of this reaction, that's a good thing because they're going to be used to help release the oxygen from the hemoglobin. All right. Another factor is bisphosphoglycerate. And what do I want you to know about this? Keep in mind, note, red blood cells don't have mitochondria. Therefore, no aerobic respiration. Only anaerobic. So, bisphosphoglycerate, no, red blood cells don't have mitochondria, therefore no aerobic respiration. However, there is anaerobic respiration. And BBG is a intermediate in anaerobic. BBG causes oxygen to be unloaded. Good. And good. All right. So is that 4? Yeah, that's 4. So right here. Okay, now, blood gases and respiratory rate. Systemic arterial blood, the pressure of oxygen, we already know, is 95 in normal conditions, 95 millimeters of mercury. Carbon dioxide is 40. You can look back at the pictures that we've drawn to show this. And then the pH of blood likes to be, where's my purple? Purple. 7.4. 7.35 to 7.45. Okay. Rate and depth of, all right, so those are some facts. Let's see. Rate and depth of breathing maintains these values. How? Okay. And then we have 2 more questions. How does hyperventilation fix acidosis? And how does hypoventilation fix alkalosis? The end, but see notes above. Alrighty. For this first one, why is pH... rate and depth of breathing maintains these values. How? Well, basically it's maintained because the brainstem receives a stimuli from chemoreceptors that monitor. So how can we write this? Brainstem. Remember that brainstem, the pons and the ventral respiratory group and the dorsal respiratory group are the ones that maintain and balance your breathing rate. So the brainstem receives stimuli from chemoreceptors. That monitors the composition of blood. And cerebral spinal fluid. Because the cerebral spinal fluid is what's going to flow over the brainstem via the pH. This is the driving force. pH is the driving force of the rate and depth of breathing. All right. Next is how much carbon dioxide levels, and then last, oxygen levels. All right, so what do I mean by these? Okay, so the brainstem receives the stimuli. It finds out the composition of the blood, like the oxygen levels, CO2 levels, and the pH. The most important is the pH, because if the pH changes, then, and it becomes too acidic, let's think about this. If your blood becomes too acidic, that means, here's a blank piece of paper, that means you have a lot of hydrogen ions, okay? So too acidic, pH is less than 7.4. Let's say it's 7.1. That's too acidic. So you know that you have too much CO2, all right? Too much. Because when CO2, because we know, this equation I keep showing you all the time, if you have a lot of this, you're going to have a lot of this, all right? So, actually, so how could you fix this? How could your body fix this? If your pH is too acidic, then you need to get rid of CO2. And how can you get rid of CO2? You hyperventilate. In other words, you blow off a lot of CO2. Blow it off. So then these hydrogen ions can be used up, all right, and we can reverse this equation. The reverse is for alkalosis, okay? So, but that can help you with these two questions. But I want to back up. Why is pH the most important factor to regulate the rate and depth of breathing? This is important. So, hydrogen ions don't cross the blood-brain barrier. Very easily. However, CO2, carbon dioxide, does. If too much CO2 crosses, therefore an increase in hydrogen ions, because once it crosses, it breaks down. All right, so now you have a lot of hydrogen ions, and this is what's detected by the brainstem, because there are no buffers to use up the hydrogen ions. Therefore, you have a drop in pH. So that's why the pH is the driving force. I'm going to leave you with these two questions. How does hyperventilation fix acidosis? The last one, how does hypoventilation fix alkalosis? Whew, Lordy, I'm tired. And I know you're tired of listening to me. Just take it in small chunks, and hopefully this helps. I would love your feedback. So I would know what you thought of this. Okay, bye guys.